Not like in a karyotype, a serial arrangement of chromosomes of a cell is known as a karyotype or karyograph. The first step in karyotyping is spreading of metaphase chromosomes and a slide and binding of them by a specific banding technique. Chromosome can be recognized in preparation of metaphase cells by their size and shape and by pattern of light and dark bands. Convert the chromosomes in many well spread countable metaphase spread and selected for photography. So first we will be needing a banding technique. We will be spreading metaphase chromosome. Metaphase spread is prepared. Okay. So a serial arrangement of chromosomes of a cell is known as karyograph or karyotype. The first step in karyotyping is spreading of metaphase chromosomes of slide and banding of them by a specific banding technique. Chromosomes can be recognized in preparation of metaphase cells by their size and shape and by pattern of light and dark bands. Convert the chromosome in any well spread countable metaphase and selected for photography. Photographs are taken with a black and white or color film uploaded in a camera attached to the microscope. Photographs are taken with a black and white or color film uploaded in a camera attached to the microscope. Then 8 by 10 enlargement of photographs are made. From the photographs, individual chromosomes are cut and removed and are arranged according to the International System of Human Cytogenetic Nomenclature. This systematic arrangement of chromosome is from a cell of group of homologous pairs according to their length, position of centromere and spacing binding pattern is known as karyotyping. The nomenclature, the chromosomes and their band types have been numbered by a universally accepted nomenclature ISCN, International System of Cytological Nomenclature. The short and long arms of each chromosome are referred to as P and Q respectively. There are 23 pairs of chromosomes in human complement, 22 pairs of autosomes and a pair of sex chromosomes, XX in female and XY in male. The members of each individual pair are identical and are called homolog. For karyotyping, the chromosomes are arranged in homologous pair in descending order in length and their specific banding patterns. The pairwise numbered starting with the longest pair as pair 1 to 22. The X and Y chromosomes are given traditional letter designation. So human chromosome fall into 7 major groups A, B, C, D, E, F, G and having similar features. Group A consists of chromosome number 1, 2 and 3. Group B consists of chromosome number 4 and 5. Group C consists of 6 to 12 and X chromosome. Group D consists of 13 to 15. Group E consists of 16 to 18. Group F consists of 19 to 20. Group G consists of 22 to 20, 21 to 22 and Y chromosome. Types of chromosomes are seen in human karyotype: metacentric, submetacentric and acrocentric. Group A: metacentric, submetacentric and acrocentric. Group A: longest and human chromosomes and have metacentric and only moderately submetacentric centromeres. 1 and 2 are metacentric and 2 is submetacentric. Group B: large chromosome with submetacentric centromeres. The pair with larger short arms should be designated as 4. A medium-sized chromosome in group C. 6, 7, 8 and 11 have more centrally placed centromere than pairs 9, 10 and 12. Group D: larger acrocentric chromosomes and have extremely short arms. Then group E: small submetacentric chromosomes, arms become progressively shorter. Group F: very short metacentric chromosomes. Group G: very short acrocentric chromosomes. Then Y chromosomes usually larger than the other G chromosomes. Long arms of Y chromosome tend to be parallel rather than diverging as do those of other group G members. Then X chromosomes about the size of chromosome number 8 or intermediate in size between 7 and 8. Then Down syndrome. It is an important instance of trisomy in human being. Here is a 21st chromosome is trisomy. It was first described by Lingen, a British physician, in 1926. The incidence of this syndrome in the general population is roughly 1 in 600 live births. 600 ഒരാൾക്ക് ഡൗൺ സിൻഡ്രോം ഉള്ളതായിട്ട് കണക്കാക്കപ്പെടുന്നു. Manifestation of Down syndrome include mental retardation, short stature, then broad skull and stubby hands, low simian crease on palm, short neck with excess skin at the back of the neck, then white spots on colored patch on eye called brushfield spots, a deep between first and second toe. Other cognitive and behavioral problems: delayed language and speech development, short attention span, poor judgment, slow leaning impulsive behavior. The mouth is usually open with large tongue with distinct furrowing through cardiac malformation, and the life expectancy is reduced. Sexual maturity is generally not attained. Males appear to be sterile, but few females have reported to have born children. So, in Lin joined in 1959 explained that the molecular reason for mongolism, a small autosome in the group number 21 added to the normal complement. Instance of Down syndrome is closely related to the maternal age. 21 is arising through the non-disjunction in oogenesis. In females, oocytes remain in arrested state in the early prophase of meiosis. Oocytes remain arrested in the early prophase of meiosis for long period. A period of over 40 years may permit painting of spindle fibers and result in non-disjunction. Another expansion for trisomy is related to the life period of an egg. The fertilization occurs towards the close period of viability of egg. It leads to chromosomal abnormalities in children. Then Klinefelter syndrome. Klinefelter syndrome is a genetic condition that results in a boy is born with extra copy of X chromosome. So Klinefelter syndrome is a genetic condition affecting male, as it often isn't diagnosed until adulthood. It was described by S.F. Klinefelter in 1941. Klinefelter syndrome may adversely affect testicular growth, resulting in a smaller than normal testicles, which lead to lower production of testosterone. The syndrome may cause reduced muscle mass, reduced body and facial hair, and enlarged breast tissue. The effects of Klinefelter syndrome vary, and not everyone has the same signs and symptoms. Signs and symptoms may include low sperm count or sperm, taller than average height, decreased facial and body hairs, enlarged breast tissue or gynecomastia, small testicles and penis, loose extrae, weak bones, increased belly fat. So Klinefelter syndrome is caused by the presence of an extra X chromosome in cells 47XXY. In some mosaic Klinefelter, some normal cells have some abnormal cells. Several conditions resulting from the presence of more than one extra sex chromosome in each cell are sometimes described. These conditions arise when fertilization happens between an egg and XY sperm. So, majority of Klinefelter syndrome child is born to mother at the age of 60. And Turner syndrome. Turner syndrome is associated with an abnormal female phenotype discovered by H. Turner and his associates in 1938. The affected females are monosomy for X chromosome, and chromosome in X is partially missing. Turner syndrome can cause a variety of medical and developmental problems, including short height, failure of ovaries to develop, and heart defects. Signs of Turner syndrome at birth including infancy, wide or web-like neck, low set ears, broad chest with widely spaced nipples, high narrow roof of the mouth or palate, palate, high, narrow roof of palate. Turn towards the elbows. Arms that turn outward of elbows. Fingernails and tonsils that are narrow and turned upward. Swelling of the hands and feet, especially at birth. Slightly smaller than average height at birth. Then slowed growth, cardiac defects, short fingers and toes, low hairline at the back of the head. Turner syndrome or X chromatin negative, which suggests the presence of only X chromosome. The karyotype is 44A XO. Frequency of occurrence of Turner syndrome per birth have been estimated about 2 to 4 in 10,000 births. The origin of the effect is probably found from experimental exceptional egg or sperm with no X chromosomes. Most girls and women with Turner syndrome have normal intelligence, developmental delays, non-verbal learning disabilities, and behavioral problems are possible, although these obstacles may vary affecting individuals. Next, study of principle and application of DNA fingerprinting. Science of DNA fingerprinting was first used by Sir William Herschel as the method of identification in 1858. In India, science of fingerprinting was discovered by chance during a murder investigation in Jabalpur in 1897. Alec Jeffrey invented the DNA fingerprinting technique at Leicester University, UK. Dr. V.K. Kashyap and Dr. Lal Singh started DNA fingerprinting in India at CCMB, Center for Cellular and Molecular Biology, Hyderabad. DNA fingerprinting, also called DNA typing or DNA profiling, is a technique of determining nucleotide sequences of certain areas of DNA which are unique to each individual. Each person has a unique DNA fingerprinting, unlike conventional fingerprinting that occur only on the fingertips and can be altered by survey. A DNA fingerprinting is the same for every cell, tissue, and organ of a person. It cannot be changed by any non-treatment. The ideal way to distinguish an individual from other people would be this for her entire genomic DNA sequence. So, principle by their difference about 0.1% of or 3 into 10 raised to 6 base pairs out of 3 into 10 raised to 9 base pairs provide individuality to each human being. Human genome possesses numerous small non-coding but inheritable sequence of bases which are refracted many times. These sequences occur nearly to by their difference about 0.1% of base pairs provide individuality to each human being. Human genome possesses numerous small non-coding but inheritable sequence of bases which are refracted many times. These sequences occur near to telomere, centromere, Y chromosome, and heterochromatic areas. The areas with which sequences of bases repeat several times is called a repetitive DNA. They can be separated as satellite from bulk DNA during density gradient centrifugation and hence called satellite DNA. In satellite DNA, repetition of bases is in tandem. So, depending on the length, sequence composition, and number of tandemly repetitive units, satellite DNA have subcategories like mini satellites, microsatellites. Satellite DNA show polymorphism. The term polymorphism used when a variant at a locus is present in which a frequency of more than 0.01 population variations occur due to mutations. While mutations in the genes produce alleles with different expressions, mutations in non-coding repetitive DNA has no intermediate impact. These mutations in non-coding sequence have piled up with time and form the basis of DNA polymorphism. Variation at genetic level arises due to mutations. DNA polymorphism is the basis of genetic mapping of human genome as well as DNA fingerprinting. DNA polymorphism. So short nucleotide repeats in the DNA are very specific in each individual and vary in number from person to person but are inherited. These are variable number tandem repeats or VNTRs. They are also called as mini satellites. Each individual inherited these repeats from his or her parents, which are used to be used as genetic markers in a person identity test. For example, a child might inherit a chromosome and 6 tandem repeats from mother and some tandem repeats at times homologous chromosome inherited from father. One half of ENTR alleles of child resemble that of mother and the other half with that of father. Procedure: the DNA is extracted from the nuclei of WBC or spermatozoa or the follicle cells of hair that cling to the roots of hair that have fallen or have pulled out. The DNA molecule is first broken with the help of enzyme restriction endonuclease chemical knife that cut them into fragments. The fragments, quick recap of this step. The DNA fingerprinting process. Blood sample, DNA is extracted from blood cells. DNA is cut into fragments by restriction enzyme. The DNA fragments are separated into bands during electrophoresis in an agarose gel. The DNA band protein, the DNA band pattern and the gel is transferred to a technique known as Southern blotting. Once again, blood sample, DNA is extracted from blood cells. DNA is cut into fragments by a restriction enzyme. The DNA fragments are separated into bands during electrophoresis in an agarose gel. The DNA band pattern in a DNA in gel is transferred to a technique known as Southern blotting. The DNA probe binds to specific DNA sequences on the membrane. Excess DNA probe is washed off. At this stage, the radioactive or chemoluminescent probe is bound to be DNA pattern on the membrane. So, X-ray film is placed next to the membrane to detect the radioactive or chemoluminescent pattern. The X-ray film is developed to make visible the pattern of bands, which is known as DNA fingerprint. DNA also contain ENTR. The fragments are repeated according to size by gel electrophoresis. Then fragments of particular size showing ENTRs are multiplied through PCR technique. They are treated with saline alkaline chemicals to split them into single stranded DNA. The repeated fragments of DNA when used for hybridization, radioactive DNA probes having repeated base sequences complementary to possible ENTRs are poured over the nylon membrane. Some of them will bind to the single stranded ENTRs. The method of hybridization of DNA with the probe is called Southern blotting. After the name of the inventor, the nylon membrane is washed to remove extra probes. And X-ray film is exposed to the nylon membrane to mask the places where the radioactive DNA probes are bounded to the DNA reagents. This place is visible to mark the star band when X-ray film is developed. This is known as autoradiography. So the dark bands on X-ray film represent the DNA fingerprinting. Applications: individuality, like skin fingerprinting, like dermatoglyphics. DNA fingerprinting can also be distinguished a human being from another with the exception of monozygotic twins. Paternity or maternity disputes. DNA fingerprinting can identify the real genetic mother, father, and offspring. Human lineage. DNA from various probes are studied to find out human being lineage. And hereditary diseases. The technique is used to identify genes connected with hereditary diseases. Then forensics. DNA fingerprinting is very useful in the detection of crimes and legal pursuits. DNA fingerprinting have proved the human bomb was the real murder of Sri Rajiv Gandhi, Prime Minister of India. Paternity, maternity disputes, individuality, human lineage, hereditary disease, forensic, sociology. It can be identified in racial groups, the origin, histological migration, and national geography is a study of migratory species. Then kymograph, working principle and applications. Kymograph is an apparatus which provides uniformly distributed writing surface that is drawn on which traced paper is placed, and the glazed paper is covered with a layer of rate of movement of kymograph can be changed. So Sherrington recognized a recording drum. The apparatus has two keys which is connected to resolving pulleys. The speed can be changed by changing the gears, and there will be two terminals for electrical connection. There are two leveling screws for electrical connection. There are two screws for the apparatus. So myograph stand. It is a triangular base and base must be away from the drum X block. The depression on the X block must face upwards. An isotonic muscle lever. The downward movement of the lever must be arrested by the screw. There will be small height to record the change in the length of the muscle. The tension of muscle is kept constant. An isometric muscle lever. The level in the kymograph is used to record the tension produced during muscle contraction. The length of the muscle contraction will be constant. 4. Frog board of cork board. Will be used to keep the frog in the board on the nerve. Will be resting on the board. A large layer of cork is placed above rectangular wooden piece of board. Femur clamp or muscle grip. Femur with muscle is kept in between the femur grip. Then spring key. The key is always included in the second circuit or start circuit to stimulate the nerve, and the key is kept open. Stimulating electrodes. The red terminal is positive and the black is negative. The unit wire serves the purpose of step down transforms. It can be used instead of 2 voltage terminal. Then types of current: galvanic or direct. It is obtained from battery on low voltage terminals. This made shock will be greater than brake shock in the case of galvanic current. Usually galvanic current is not used. So induced current. Doughboy sphere mount in torrent. The plates are primary compared with the iron wire. You could see a picture of kymograph. Pointer, drum, motor upon which upon wire are wound. Secondary keel composed of otherwise connected to 2 terminals. Centimeter scale for measurement. Pointer. It is used to read the distance of secondary coliform from the primary coil. It is based on the principle of electromagnetic induction and it is used to read the electric shock. In this case, the brake shock is greater than mask shock. The length of current is inversely proportional to the distance between the coils. We get only a single shock from induced current. Higher voltage terminals using induction by increasing the number of turns of coil, the strength of current is increased. The disadvantages of induction is that qualitative work can be done using this method. Then electrostimulator. Qualitative work cannot be done with this apparatus. Then Faradic current. Quantitative material is also induced current. We will get reflector shock from cardiac output. The Neff's hammer or autonomic make and brake hammer. The vibration changes from 40 to 60 per second. When any blood is centrifuged or RBCs are separated and settle down, plasma is seen as a clear fluid. Several RBC represent packed cell volume. It is referred to as hemomount. This can be calculated by different methods. Estimation of hemoglobin of blood using hemoglobinometer. Introduction. Hemoglobin is the main constituent of RBC and carries out the important functions of gas transport. Hemoglobin consists of 2 components, heme, iron plus porphyrin, and globin, polypeptide chain. Defects in hemoglobin are called hemoglobinopathies. Sickle cell anemia, for example, there are different methods to determine the hemoglobin concentration. Here we will be discussing about Sahley's method. Sahley's method or acid hematine method to determine the percentage of hemoglobin in the given blood sample. Hemoglobin is converted into acid hematine by the action of HCl. This gives a brown color and its intensity depends on the hemoglobin concentration. The acid hematine thus formed is diluted with 0.01 normal HCl or distilled water until its color matches exactly with that of the standard, who's fixed in the configuration block. Comparator block. The hemoglobin concentration can be read elastically from the calibration tube. Apparatus and reagent 0.1 normal HCl, Sally's hemoglobinometer, Sally's pipette 20 ml, surgical blade, lancet alcohol pricking needle, and spirit swab. Hemoglobinometer. It was a box which contained comparator. It consists of a box having one slot for Sally's hemoglobin tube and two non-fading standard brown tinted glass pieces for matching the color of diluted blood. The translucent white glass is fitted at the back, providing uniform illumination for matching the colors. Sally's tube, hemoglobin tube. It is a graduated tube having markings on two sides. On one side, GM percentage, GM per 100 ml from 0 to 24, and on the other side 0 to 70%. Gram percent means SP in gram per 100 ml of blood. So simple percent means percentage of hemoglobin in relation to normal value of which is it converted to 100%. Hemoglobin pipette, it is a single pipette having a capillary tube in it. It is connected with mouthpiece through a rubber tube. There is a marking on the pipette indicating 20 cm, 0.02 mm only. This sample glass rod used to mix the blood with acid or distilled water. So simple rubber dropper to add acid or water dropwise. So procedure: with the help of a dropper, take n by 10 HCl in the Sally's tube up to mark 20%. Then under and condition, give a bold prick on the tip of the ring finger with the help of a pricking needle or lancet. Wipe off the first drop of blood and suck the blood from the second drop into the HP pipette into the mark 20 ml. Wipe the tip of the pipette with the help of a cotton to remove excess blood around the tip. Blow out the blood from the pipette into an acid and a Sally's tube and rinse the pipette with the same. And mix the blood with acid using a stirrer and wait for 10 minutes. Dilute the mixture by adding distilled water drop by drop with stirring until all of the mixture matches with that of the standard color of the comparator. Once the color has been matched, remove the stirrer and note down the reading of the Sally's tube by taking the lower meniscus. Usually for colored solutions, upper meniscus is considered while recording the reading. But since it is a transparent colored solution, the lower meniscus may be used. Reading is expressed in HB gram per 100 ml of blood. The amount of hemoglobin present in the given sample of blood is 12.4 gram percentage. Just notice about Neubauer chamber, also known as a hemocytometer, is a microscope slide that contains a counting chamber with a grid etched into the glass. It is commonly used to count cells or other microscopic particles in a sample of fluid such as blood, urine, or cerebrospinal fluid. The Neubauer chamber is a thick crystal slide with a glass slide 30 to 70 mm and 4 mm thickness. The simple counting chamber, the central area is where cell counts are performed. The chamber has three parts: the central part and the counting grid has set.